gonna, I'm gonna touch a little bit briefly on some of my more recent work, thinking about how we can analyze occurrence of different species to estimate trends of biodiversity through time. Okay, so onto the empirical and conceptual work. So spatial dynamics are a crucial process in ecology, and this ideas of spatial ecology have really kind of been gelled together with meta-community ecology, where we're thinking about how do species coexist with one another at multiple spatial scales, right? So what promotes the coexistence of species at local scales, but as well as regional scales. And so this is an example of a meta-community. You have different types of species coexisting with one another in individual locales. You also have the movement of the species from one side to the other, and together you have this meta-community. In meta-community ecology, at least back in 2011, like right before I started my PhD, proposed that there was three main processes that structure meta-communities. These were the dispersal, so we're talking about the movement of organisms, particularly in dispersal. The other one is competitive equivalence, right? So are species equivalent to one another, or are they dominant competitors or not? And how much environmental heterogeneity there is in the landscape. And so when I started thinking about meta-communities, this was kind of the framework that I started thinking with. But really, I wanted to think about further, how can we think about meta-community theory for food webs? And so my first step was, well, let's just modify these processes so that we can adjust some of these for food webs, right? So maybe let's get rid of the competitive equivalence and we start thinking more about the functional response. So the functional response relates how predators and prey interact with one another. And so for most of the work during my PhD, I wanted to know which meta-community processes contribute to food web persistence at local and regional scales. And I decided to do this both with empirical and mathematical and computational approaches. And in the empirical system, I was working with the bromeliad food web. So bromeliads are plants that have interlocking leaves. And so in these leaves, the plant collects water inside, and so they provide habitat for aquatic macroinvertebrates. Inside this habitat, you get lots of different organisms that live within bromeliads. And in particular, you get a food web of invertebrates where you have a damselfly larvae, which is a top predator, and it's a voracious predator and eats everything that's like you put in front of it. And a bunch of other invertebrates like mosquito larvae and tipulate larvae, they're all part of the damselfly food. Another interesting thing about the system is that so within the bromeliad, you can think about bromeliad as a single patch, and these organisms live there within the larvae stage, but then they emerge as adults, and then they move within, like between the different bromeliads. And then as adults, they oviposit into new bromeliads. So you have the movement, both you have a contained food web and you have the movement of organisms between patches. So I went down to Brazil, to the Restinga, the Jurubachiba National Park, and I collected data on these individuals. And one of the things I was interested in was, what structures the persistence of the predator, and why focus on the predator? Well, we know that the damselfly presence has strong consequences for the entire community. And sites that have this damselfly or not have very distinct abundance distributions. And so the presence of the damselfly gives us a good idea of what's happening to the rest of the community. And in addition, has this simplified but food web component. Okay. So within these processes that structure meta-communities, trophic meta-communities, I asked three specific questions. The first one was thinking about at the local scale within one bromeliad, do differences in body size of prey affect the local food web stability? So here I'm thinking about what's happening at the stability within a single plant. Then I wanted to really incorporate this meta-community approach and think about whether different species within a food web had different dispersal rates, right? So thinking about the meta-community, and what were the consequences of just different dispersal rates? And finally, can we actually extend the meta-community ecology to fully encompass food web dynamics? So first I want to delve into this functional response axis and think about the differences in body size and the stability of the food web. And this is a project that I did during my PhD with Diane. So something I haven't told you about the bromeliad food web yet is that there's a wide spectrum of body sizes for the prey. We have some of the prey of the damselfly, which are very small, and very, very large prey like tipulate larvae. And so we have this interesting system with this body size gradient. And so what I did is I did feeding trials with the predator with each of the different species. So here you can see, for example, a photo of the damselfly with one of the tipulates in the container, and I counted how many tipulates were eaten, and the damselfly would just like eat the tipulate, and the oligochaetes they're like spaghetti. They're like slurp the whole thing. Anyway, so for each of the species, I presented different numbers of individuals to the predator. So here on the x-axis, we have the number of prey offered, right? So how many prey I put in the container. And on the y-axis, we have the number of prey consumed. And what I was trying to do is fit this functional response, that is the relationship of how many prey were consumed versus how many prey were offered. And we were able to fit this functional response with this equation here on the top left, where there's two key parameters that we care about. First is the attack rate. So how I can think about it as how often the predator goes and attacks and tries to get prey. And then the handling time. So how long does it take the predator to search, digest, handle its prey? And the thing, because the predator, there's a point where it saturates, right? There's a point where it can't really eat everything that you put in front of it. And that represents this kind of saturation point in the curve. So one of the main things I found was that this saturation curve, this functional response, changes as a function of the body size of the prey. So I fit the attack rate and the handling time as a function of body size. And I found that as you increase the body size of the prey, the attack rate goes down. And as you increase the body size of the prey, the handling time goes up. I was like, great, that's awesome. Now we can use these relationships to understand the persistence of the food web. So I developed a predator-prey food web model where the abundance of the prey was determined by the abundance of the prey in the previous time step, plus new prey added via population growth based on the prey body mass, minus competition based on the prey body mass. So two prey species that were closer together in body size competed more strongly than two species that were further apart, minus predator consumption based on the prey body mass. And this is where I parameterized with the experiments. The equations for the predator were very similar. So the predator abundance was determined by the predator abundance in the previous time step, plus new predators added via the consumption based on all of the prey body mass that was available, minus mortality. Okay, so I started with one prey, one predator. So on the x-axis, we have time, so the simulation time. On the y-axis, we have abundance. We have our predator abundance in red and our prey abundance in blue. So the first thing that I found was that when the prey is very small, basically the attack rate of the predator is very high. So the predator is going to find every single prey, drive it down, and drive itself extinct. At the opposite end, when the prey is large, the handling time is very long. So the predator cannot obtain enough energy. It drives itself extinct because it doesn't have enough energy, and the prey is allowed to persist. So it's really only at this intermediate body sizes that both the predator and the prey are able to persist through time. I was like, great, this is, but this is only one species, and I was thinking about a food web, right? So let's increase the number of prey available for this predator. So from previous studies, we knew that the stability of a food web depended both on the diversity and the body size ratio between consumers and resources, between predators and prey. And basically, when this ratio is small, smaller food webs tend to be more stable. And as the ratio increases, larger food webs tend to be more stable. But I wanted to break down this, this ratio because not all of the interactions within a food web would be the same ratio. So I started increasing prey diversity. And one of the things that I ran across was the fact that as I increased the diversity of my food web, as I added more prey, the number of parameter combinations that I would have to search through increased really fast. Like here on the y-axis, this is on log scale. And it became to a point where it was computationally unfeasible to understand the persistence of the food web as we increase food web size so much. So I decided to use this MCMC Monte Carlo strategy for searching parameter space, where I was able to reduce parameter space by searching what were the interesting areas where something happened. And in particular, I was interested in the persistence of the predator. And this allowed me to go from 10 to the 20 parameter combinations to 20 to the 4, a much more manageable problem. Okay, so let's start increasing prey diversity. So here on the x-axis, we have the body mass of prey number 1. On the y-axis, we have the body mass of prey number 2. In yellow, it's the areas where all 3 species persist. And in purple is the areas where at least one species doesn't persist. The lower triangle is basically the mirror image, which is why I'm not showing it. So following what I had just found with one prey, when the body mass of prey number 1 is intermediate, I find that there's persistence of all 3 species. But the interesting thing is that it didn't really matter what the body size of prey number 2 was, as long as it was larger than prey number 1, which is what we see here. Okay, so we know that what starts to matter is just the body size of the smallest species. So I started to increase diversity. So here we have prey diversity on the y-axis. So I flipped things around, and we have body mass of the smallest prey on the x-axis. And what we find is that the predator persistence is constrained with the body size of the smallest prey, and that body size has to be intermediate. But the range of values actually changes with diversity, which is kind of cool. So the range of values increases and then decreases as we increase diversity. And you may think, well, why? And one of the reasons is due to Jensen's inequality. So I'll walk you through what Jensen's inequality is. So imagine we have 3 species with 3 body sizes, and we calculate the attack rate for each of these 3 species, right? So we calculate the attack rate. And then we calculate the average attack rate for these species. Now, there's another way of doing this order of operations is say, well, let's calculate the average body size of all these 3 species. And then we calculate the attack rate of this average body size. And Jensen's inequality tells us that when this relationship is not linear, so the relationship between body size and attack rate is not linear, the average attack rate is not going to be the same as the attack rate of the average body size. And so this is just the exact same thing I just told you, but in graph form. So where the average attack rate is higher than the attack rate of the average body size. And this happens because this relationship is not linear. So when we increase prey diversity, so here we have on the x-axis the attack rate of this average body size. And here on the y-axis, we have the average attack rate. So when prey diversity is 1, these 2 values are the same. Great. Now, as we increase diversity, the average attack rate, which is what the predator is experiencing, increases much faster and then decreases again. And this is part of why we see this increase and then decrease in the persistence of the predator. And so overall, what we found was that prey diversity had this unimodal relationship with predator persistence. And it was mediated by this Jensen's inequality on the body size, like body size attack rate relationship. Okay, so in terms of thinking about the functional response, what's happening at a local scale, I told you that the predator persistence was maximized at intermediate prey body sizes, that only one prey species really constrained the predator persistence as long as that species was intermediate of intermediate body size. And then the prey diversity had this unimodal relationship with predator persistence. So I wanted to continue and then think about this in terms of dispersal, right? So what's happening? Do different species in a food web have different dispersal rates? And what are the food web consequences for these differences in dispersal rates? And again, I did this work with Diane. So I was down there in Brazil and I collected individuals from 10 different sites. But I spread out my sites so that the first few sites were very close to each other. And then as I increased the sites, I increased the distance between the sites. And the idea was that we could look at the relatedness of individuals between sites to get an idea of how much movement there is between the sites. And by increasing the number of sites, like the distance of the sites, we could get some sort of dispersal kernel. So for each site, I collected 10 to 20 individuals from each species. I extracted the DNA for each individual, I genotyped individuals, and then I estimated the dispersal between sites. Okay, so I'm really glossing over the methods here. And initially, as all science goes, it was supposed to be for a food web, but we ended up with the predator-prey pair. So I was able to estimate dispersal rates for the damselfly predator and one of its prey, the tipulate larvae. And the cool thing is I had this dispersal rate, but I also had the feeding links between these two species. Now, based on body size alone, we would expect the damselfly to have a larger dispersal rate at all distances than the tipulate. But that is not what we found. What we found was that the dispersal rate of the tipulate was always higher. So here on the x-axis, we have the distance, and on the y-axis, we have the dispersal rate that we estimated between each pair of sites. And here the inset is basically filtering out everything that the distance was above 0. Because basically the 0 distance was, the same sites. And what we see is that for the tipulate, the dispersal rate is actually higher at most distances, which was interesting. And so we were thinking, well, what are the consequences for the predator-prey pair of this result? And one of the things I was wondering is, well, can the observed asymmetry in dispersal account for occupancy patterns that we observe in nature? And I say dispersal asymmetry because the tipulate disperses more than the damselfly. But based on body size, we would have expected the opposite. So I had my functional response from the previous study that I told you about. And I was able to use this again in a predator-prey meta-community model. So here I did a very similar model where we had the prey abundance be determined by the prey abundance in the previous time step plus new prey added via population growth minus predator consumption plus immigration minus immigration. And similarly for the predator, we had that the predator abundance in the previous time step was determined, the predator abundance was determined by the predator abundance in the previous time step, plus new predators added via population growth. So this was just acknowledging the fact that the predator can consume other type of prey, not just the tipulate, and it could have an intrinsic growth rate based on eating something else, plus new predators added via consumption, minus predator mortality, plus immigration, minus immigration. And note that both the predator consumption, the immigration, and immigration were all parameters obtained from the empirical data. So there's multiple things that we could have expected. And so this is kind of like the expectation of the results. So we could have, so if you think about each of this grid as a little site, we could have no persistence of any species. We could have a case where only one species persists, either the predator or the prey. We could have a case where both species persist and they always occur. Or we could have a case where they both persist and different in occupancy, different in their occurrence. And so what we found, so we did simulations. And one of the things I wanted to test was this, this asymmetry in dispersal, right? So we have the observed asymmetry, which is that the prey disperses more than the predator. And I also tested the reverse, a scenario which we would have expected, where the predator disperses more than the prey. So what we found was that with the observed asymmetry, we're more likely to find the prey and the predator that survive with different occupancy. Whereas in the reverse scenario, we're more likely to find the predator and the prey with the same occupancy. And when I went back to look at what's the observed occupancy that we see, we have this difference in occupancy between the predator and the prey. Now, this is just one data point. I could have sampled many more sites, but at the meta community, we observed this difference in occupancy. And one of the reasons that we observed this was that allowing the predator to consume other prey makes the difference in occupancy possible, right? Like if the predator only persists on the tipulate, this differences in persistence would not even have been possible. But the reason why it's more likely in the observed scenario where the prey disperses more than the predator is that in the case when the predator has higher dispersal, these differences in occupancy are only possible when the predator has a low growth rate. So basically in the observed, the prey having higher dispersal rate, there's more parameter space where the predator the predator could have a low and a high growth rate and still have these differences in occupancy. Okay, so in summary, in terms of dispersal, I told you that tipulates have a higher dispersal rate than odonates, but also that this observed dispersal produced higher persistence and difference in occupancy than the reverse. So I wanted to continue thinking about this meta-community processes. Particularly as we're thinking more about food webs. And I was thinking about this environmental heterogeneity. And environmental heterogeneity assumes, that the species have the same breadth of abiotic niche, right? But in reality, when we're thinking about food webs, when we're thinking about interacting species at different trophic levels, this may not be true. And so environmental heterogeneity doesn't really work when we're thinking about food webs. And so the other aspect of my work, I wanted to think about, well, how can we extend meta-community ecology to fully encompass food web dynamics? And this is a work that I did with a great group of people in a working group. And we were really, what we wanted to do was highlight, assumptions where competitive meta-community theory really didn't fit for food webs. And we wanted to propose a way forward. And so some of the assumptions that we highlight were very simple, like they're very obvious in retrospect, but competitive meta-community theory and many of the competitive meta-community models assume that species that are interacting experience the environment at the same spatial and temporal scales. But we know that, in food webs, this is not necessarily the case. We may have cases where a more motile predator may experience the environment at larger scales and therefore experience different levels of environmental heterogeneity. Another assumption was that species move naively, right? But instead we know that species make decisions, active decisions about where to go. And finally, it assumed that movement didn't vary between species. So many meta-community models would have the same dispersal distance, but varying the rate. But in reality, we know that the distance actually varies. The scale of movement can vary with body size, but it can also vary with trophic position, particularly in marine food webs. So we wanted to propose these things called spatial use properties, which are population level properties that reflect how species use space based on their traits, behavior, body sizes, and other attributes. And there were 5 spatial use properties that we identified that were important for food webs. These were the abiotic niche and dispersal scale, which I've already told you a little bit about from competitive meta-community theory, but we also wanted to include things like foraging scale, migration scale, and spatial information processing. So I want to briefly touch on these foraging and migration scale and spatial information processing and why we thought it was important to include them. The main point is that there's different forms of movement, there's different types of movement like foraging, migration, and dispersal. First, they occur at different spatial scales. Some of these movements and actually migration may even be at a larger spatial scales for some species, but they connect different types of habitats at different distances. And their frequency is also very different. So for example, foraging may occur very often within an organism's lifetime. Migration may occur seasonally, while dispersal only occurs once in a lifetime. And the other thing is that, for example, if you have a migratory species, the nutrient pulse that they bring to their incoming food web is very high. So the consequences for community dynamics of these different types of movement is quite different. The other one that we wanted to highlight is this idea of spatial information processing, which is a big umbrella of the idea that, first of all, species sense their environment and that they decide where to go. And these sensing of the environment, the decisions about where to go affects all 3 types of movement, so foraging, migration, and dispersal, and that they may differ between species that are interacting with one another. So predators and prey may sense their environment in different ways and may have different abilities to decide where to go, which has consequences for community dynamics. And so really what we wanted to highlight here is that the way that spatial use properties will affect food web structure depends on how these properties differ between species in a food web, right? So, you know, if all of these things are the same between predator and prey, they probably won't make much of a difference. But if they differ, potentially they will. And so the first thing that we wanted to do as a subsequent study from this is thinking about, well, can we actually characterize this? Can we actually find whether, you know, foraging, dispersal, and migration differ between species in a food web? Do they covary with one another? Like, how can we simplify this problem when we're thinking about real meta communities? And so this is a project that we did with Sam Strauss. We recently just finished it. And our main question was whether these types of movements, so like foraging, dispersal, and migration, covaried with one another, because if they do, that simplifies our problems. We don't have to worry about them very much. And what would be possible constraints? Like, do they covary equally across taxonomic groups? Do they covary equally across trophic levels and things like this? And the short answer is that they do. There's exceptions. So, and the exceptions are what kind of make it interesting. So, you know, this is some of the work that we've taken after that initial working group. I also, you know, wanted to briefly highlight some of my other work thinking about meta-community ecology, because most of my work has been thinking about food webs, but I've also done work in the competitive, like thinking about competitive interactions. In particular, I've worked on thinking about how we can incorporate local coexistence theory with spatial dynamics. And most recently, I've been very interested in, okay, we have all of these meta-community theories. How do we actually link this with empirical data? What do we need? And how can we use empirical data? Like, how can we understand meta-community processes from empirical data? And so I've taken kind of like a virtual ecologist type of approach, doing many simulations and trying to link these meta community processes to signals like descriptive statistics and things that we see from the empirical data. And so in the last 10, 15-ish minutes, I want to tell you a little bit about some of the work that I've done during my postdoc and now in my new position, mostly because it's a little bit different. And a lot of my future work has been based on this more recent work. And I was funded by the Librero Fellowship for this more recent work. So museums have an enormous and vast amount of data. Museums are full of boxes and drawers of different specimens collected at different points in time for different species. And recently, there's been strong digitization pushes to have digital data for all of these specimens that we have stowed away in museums. Here, this is a contraption of like a bunch of different cameras that can take this like really cool 3D photos of this specimen. So you can get all sorts of like morphological type of data, but more basically as well, we have information about who the species are, where they were collected, when they were collected, and by whom. So just, digitizing the labels itself. And in addition to this like strong digitization pushes, citizen science has exploded in the past decade, and citizens and students and all sorts of people are collecting data, nature data using apps like iNaturalist, Bumblebee Watch, eButterfly, and these have become a much a more common way to collect biodiversity data. And so if I took this slide like 6 months ago, and I'm sure it has increased since then, but if you look just at the number of occurrences, GBIF alone has over 120 million occurrences of different species. And these points are, not everything is clean and not everything is correct, but the amount of data that is available through this community science and the digitization pushes is pretty impressive. But one of the main problems is that this occurrence data changes a lot through time and space. So for example, we have observed that the amount of data available for bees has increased tremendously in the past 20 years. And if you analyze this data on its own, just raw, you would assume that species are increasing through time, which we know that may not actually be the case. Similarly, when we look at this data across space, we would assume that cities are our most biodiverse hotspots, when in reality, it just seems that cities are the most convenient place to collect data. And so while these data might be very useful, we have to think very carefully about how we analyze this data. Otherwise, we may have very biased inferences about biodiversity. And so I've been working a lot thinking about how we can use occupancy models to analyze this data. And I'll tell you very briefly what an occupancy model is and how we can use it. So when you, in the most traditional application of an occupancy model, you would set up a few different sites, and then you would go and visit them in a couple of intervals. And then let's say we have 4 sites right next to a farm, and we go and see whether this bumblebee is present in the different intervals. Now, in this case, for example, in this site, in this time interval, we didn't see it. And that's because our ability to detect species is not perfect. And every time we go to the field, we're not going to observe we're not always going to observe species necessarily. Now here it's very easy to say, well, we've observed it 2 times, for sure it's a misdetection. In this particular site, we've never observed it, but our ability to detect species is actually pretty good. Like 2 out of 3 times, we most often see it. So this is probably a site where the species is absent. Now, in a case where our ability to detect species is worse, we may only observe the species one time every time we go see it. And so in this site where we don't see it, it's harder to figure out whether the species is truly absent or whether it's a misdetection. And so really what occupancy models do is that they help us disentangle between, these two different types of zeros, like is the species absent or is it just a misdetection? And these types of models, I think can actually be applied to these unstructured data, but we need to figure out how. And so some of my work during my postdoc has been thinking a lot about, how can we properly apply these models to this occurrence data, right? It would be a shame to not use these data just because it has all of this different, biases in space and time. So we have to control for this. But this is a very rich data set that we can use to understand changes in species distributions. And one particular problem that is interesting is thinking about the absences, right? So I told you about occupancy models and how they have to disentangle between these true absences and misdetections. But when we're working with GBIF, historical records, citizen science, all we have is the presence. We don't really know when people, we don't really have the absences. So some of the things we have to do is actually infer these absences. Naively, we may think that, we'll just fill in the zeros, right? Well, like this site time interval combinations, they're just zeros. But really, it may be the case that the site was not even visited in that time interval, or that it was visited and it's not in the database, or that it was visited and a B wasn't found. And so we have to distinguish between these types of zeros, because modeling the sites that were never visited wouldn't necessarily be correct. Another type of question that I've been thinking about is, well, how does collector behavior affect both the type of data that we get and how we should analyze it? And so there's people that, they may be doing a taxonomic revision, and so they're specifically target some species. You may have some ecologists that they go to a site and they just want to assess diversity of everything within the site. So they do a community type of sampling. And so the people who are targeting individual species, they may not even visit. So for example, in this particular case, this targeted species sampler is interested in the yellow beetles, orange beetles. So they may not even visit sites that don't have orange beetles. And the sites that do have orange beetles, they only collect the orange beetles. Whereas the community sampler, goes to all of the sites and collects all of the species. And so their resulting collection, may look something like this, but the data that we get looks like this. We don't know what the underlying community is, and we don't know what type of collector different people are. And it gets more complicated when we start binding together different types of visits and collections. And so my work here has been thinking about, let's simulate data like historical records, thinking about all of these different intricacies that I've been telling you about, and then work with different modeling approaches and data processing workflows to see what is the most robust way to analyze this data and recover the trends of changes in distributions through time. The other part of my work I've been doing is actually applying these models to understand changes in distributions of different species, and in particular bumblebees. And so this is the work that we co-supervised Hannah as a master's student for. And we wanted to understand, in the past, basically what we wanted to know whether there was evidence for bumblebee declines in North America for the past 120 years, whether the responses were very species specific, and whether we could attribute changes in temperature, precipitation, or floral resource to these changes in occupancy. And the takeaway, the takeaway is that there's lots of species specific responses. Anything we do at a genus level doesn't really tell us much of the story. And so we really have to look at what different species are doing. Now, one of the things that has made us, I would say, like more relieved that the analysis is working is that the species that are known to be declining, for example, like Bombus affinis, we also find to be declining. And the species that we are known to be increasing, we also find to be increasing. And so we find that our model results are consistent with IUCN trends for some of these species. And they're consistent with the life history strategies that we know about for some of these species, like they like wet areas and things like that. So some of my next steps, thinking about what I want to do with this work. So I recently hired a postdoc, Jamie Luthwaite, and she's going to be working with the similar types of questions, thinking about butterflies. And our hope is that we can have a streamlined way to assess different types of species for Canada, as she is part of COSEWIC. Another project that I'm currently working on as well. So I've been collaborating with this group called I Dig Bees, where we have data for 3,159 bee species in the continental US. And really I want to figure out whether the distribution of these thousands of bees has been changing and what can be some potential causes of this change, particularly thinking about the effects of climate change and the pesticide use. The US has a really good detailed data on pesticide that has been used since 1995. And so we can get at this question of, have the ranges of these species changed because of pesticide use? And so that's something that I'm currently working on. And with that, I told you lots of different things. And with that, I want to thank you. Thank you so much for your attention. And I'm happy to take questions both about science, but also about other things.
Thank you so much. That's great. I'll just kick off the questions. I'll remind our viewers that you can send, you use the chat, you can raise your hand. We will recognize you. You said that your postdoc was partially funded by Liber Aero, which of course has a big science and society public-facing feel to it. Could you talk to us a little bit about the uptake your work is getting in social contexts, not just the scientific community?
Yeah, that's a good question. So in kind of like there's multiple areas of the social context. So one of the upticks has been in terms of media. So I didn't talk about this very much, but one of our analysis, thinking, simulating historical data, also included an analysis on bumblebees. And we didn't initially, try to do like a press release or anything like that, but like the university picked it up and, they quickly did a press release and we're talking to the CBC. And I was definitely, that was at the beginning of my library and I wasn't quite trained on media at that point. And so that was, I think people really care about some of these results, particularly thinking about bees. And so that, in that case, we had a lot of interest from the media on our results. In terms of other aspects, some of my collaborators, and this is part of the Librero Fellowship, which is, you're supposed to collaborate, or part of your mentorship team is also people outside of science or adjacent to science, right? So NGOs or government. And some of my collaborators here, Laura Miranda, so she works for Pollinator Partnership, and Leif Richardson, he works for Xerces. And they've been very positive about our results. I think they're quite excited. And I think the next step that we're trying to figure out is how can we distill this information to make it useful? Not just understanding, okay, well, these species are increasing and these species are decreasing, which is one goal. And for bumblebees, the gap wasn't there yet. Like bumblebees was a great test case because we know a lot of things about bumblebees. We know which species mostly are increasing and decreasing. And so it was a good test case of like, okay, are we like, is this approximately correct? And both Leaf and Laura have been excited about, the fact that we can do these analysis, across the board, which then we can export to other species that we may know less about. And the other part is that I want to do with this work is actually try to identify areas where we see most of the decline. So can we identify regions where we see many species declining? Because then that would, Laura works a lot on, prioritizing habitat for restoration. And so we find that there's particular regions that for some reason, many species are declining a lot, maybe we can do something about it. So I think they're tentatively excited and we're working on how to make it more concrete.
Audience questions. This is where being online is dangerous because I can't glare at you individually, but do we have questions for Laura? Mark. Robin is giving you powers, so hang on. There we go. Okay, Mark, I think you need to unmute yourself and then you can ask your question.
Can you hear me now?
Yes,
Yes.
great.
Oh, sorry, the option just came open there. All right. So Laura, wonderful talk, very interesting. As an insect physiologist, I love talking about insects. So I have a question for you though. I think of these meta-analyses and I look at the food webs. What happens when you expand this? Because in the bromeliads, I'm thinking you've isolated this to the invertebrate communities that are in there. But there are other invertebrates who would be, I would assume, preying on some of these species in the bromeliad community. So things like, given the climate, salamanders, I would assume some crabs, things like that. Would that have any effect on the predator-prey interactions if you incorporate those into your model?
Yeah, that's a good question. So in terms of the bromeliad system, there's also a lot of work. So here I blissfully ignored anything that wasn't aquatic, but we know that there's even other invertebrates. So like spiders also prey on the aquatic community. There's also birds that prey on the aquatic community. And there's been some really cool experiments done by some of the people working in bromeliads where they put like a fake bird right outside of the bromeliad, and then they estimate oviposition rates of different invertebrates. And basically having a bird there actually does change the behavior of some of these individuals. And so they don't oviposit quite as much. So definitely, I think including other aspects of the environment and of the food web would change our understanding of how these food webs persist. And even, this was a very simplistic simulation because at the end of the day, what I was trying to incorporate was like, can we make these experiments and actually inform these food web models in a more realistic way? Because it's great to just do models for the sake of models, but I really wanted to ground these models with empirical data. But there's other things like we can think about. There's even, so for example, there's been some cattle tank experiments with as well damselflies and chelicets. And what they find is that if you have sets of cattle tanks that are together, and one of my collaborators actually works on this, if you have a damselfly in one of the tanks, the mosquitoes won't oviposit on the tanks right next to it because they'll smell the pheromones from the predator and say, oh, this whole area is like danger. So there's some kind of like contagion of like danger as well, which are some of the other things like I didn't even like include or think about. But that definitely changes our understanding of like, what how these food webs persist. But for example, this experiments looking at the risk contagion, actually, like, they very much go to this idea of this spatial information processing, right? So clearly these organisms are perceiving the environment, not just at the individual cattle tank, but they're perceiving it at, the whole region, and they're making decisions about where to go based on that. And so I would love to include it at some point.
No, I think it's such a wonderful, elegant study. One of the things I often, when you think about meta-analysis, is like you said, this was anchored in empirical data, which I really love. And then the fact is, now can you expand that with your wonderful model system, add other factors in, and actually prove that, like, get a meta-analysis that go the other way and go say, okay, now we do the empirical experiment and we show that that's true. I think this is very elegant. I really, really like this talk, and the fact that it was based in that empirical data. Really, really nice job. And can I ask one other unrelated question, not science question?
Yes, please do.
I had to happen to look at, I stalked you on your website, Congratulations on your new position, by the way.
Thank you.
I note that you are very open about the idea that, you reflect on the fact that you don't look like the typical scientist that you when you grew up. I think you have actually a thing on your webpage or an Instagram about that. So can you comment on your experiences in getting your new position, how that came about, how you felt about that, or did you encounter any difficulties while getting your new position?
Yeah, that's a good question. So I think, yeah, for me, it's one of those things that, like, going forward, I try to ignore, but seeing, backwards, then I'm like, yeah, like, I think, so in that Instagram bit, like, they asked us to reflect, like, how did he come into science and stuff like that. And for me, like, I always knew I wanted to be a biologist since I was like 9 years old. Like, how, I have no clue, but I was fascinated with biology, and I wanted to be a scientist, and I didn't even know what that meant, but, like, that's what I wanted to be. And I didn't, I think it's one of those things that you don't think consciously. Like, I personally didn't, of realizing, at least when I was going through undergrad, that, like, I didn't see people who looked like me. And it was all, at that point that you think about it, it's like, oh, actually, yeah, like, I didn't, I did my undergrad at McGill, and I didn't see anybody who looked like me as professors or as postdocs or anything like that. And you're like, interesting. But in terms of, how that affected me getting a position and all these things, at the end of the day, I think I just have to assume that it doesn't and try my best. And it probably does. But it doesn't. So one of the aspects where it definitely does is, like, English is not my first language. And I've worked extremely hard throughout my PhD to improve my English. And even, like, before, like, improving my English has been, like, my number one goal since I was in high school. And I devoted a lot of time during my PhD on learning how to write. And I relied, my advisor, like, she was a saint because my writing was terrible. But I often, the people who I know are good writers, I'm always like, okay, can you read this? Can you read this? Can you read this? Can you give me feedback? And I've been very deliberate about making sure that I can improve my writing to get me to this point. And I was very conscious about that when I was, applying for my position. Yeah. Sorry, that was a big ramble.
No, that's fantastic. I will say one thing is I did happen to read your mentorship statement. I must say, for a person in your career, at your stage of career, I am amazed with your mentorship statements and your advice you give students in your mentorship statement on your webpage. And I would ask permission, if you wouldn't mind, if maybe we could circulate that to our department and show people what that looks like. It's a very well structured and fantastic statement. So I think, congratulations on your position. Wonderful job.
Thank
Well
you.
earned, obviously. So thank you very much. That was wonderful.
Thank you. And I just want to say, actually, so that mentorship statement, I once saw like on one of these like blog, ecology blog websites, like these are my mentorship statements. So I looked at that and I looked at other people's and I put it together and then I put my own kind of like ideas on there. So it's not original, but I wanted to put those things out there. I think that one of the things that I think was missing, or I find that sometimes is missing, is like clarifying those expectations right off the bat of, like as an advisor or as a student, like what you should expect about these interactions. And obviously those things are going to change, but, setting the tone for the conversation, I think is something that could be useful. I don't know. I don't have students yet, so I'll report in a few years how it works, but please do circulate.
Thank you very much. I'll let someone else talk now.
Shirley, please.
Hi, Dr. Guzman. Thank you so much for your fascinating talk and your share of your experience. So I just wonder, could you share any tips on how to improve English speaking and writing skills? How to get a position in academia, especially for people whose first language is not English? Because there are lots of international students or immigrants in Canada for sure. So do you have any things that you want to share with them? Yeah, please go.
Yeah, so in terms of writing, so I was very fortunate. There was a scientific writing class at UBC. Diane, my advisor, taught it. She's great. And so that was kind of like the first intro. But then, Steven Pinker has a book, and I will have to look it up and I'll send it to you, which is something about writing something. I read it and I looked at the exercises and I actually like copied some of these things, because some of the things that he explained as like connectors, like how to connect sentences, and like that's very specific detail, but like I would take his tables and like put it right next to my office to be like, okay, I need to connect to sentences. What words can I use? So his book is really good, Steven Pinker. And then I also found another couple of books. They weren't that good, but I tried them anyway. It was like scientific writing for like ESL students or something. I can look it up in my library and send it to you. And then they would have exercises. And so I actually did the exercises. Nobody like looked over them, but I tried them out just to, get at these practice of, doing things. The other thing that I did that was really helpful. And this is not so much about writing, but it was more generally about how to learn from people's feedback. There was a book called The Art of Giving Feedback. I'm very bad with names, so I can hang on. So
I think I've read that one. Yeah, I've read that one too.
So The Art of Giving and Receiving Feedback. That was amazing because it gave me lots of tips about how to communicate with my advisor and my collaborators. And sometimes I would just sit down with them and I was like, I don't understand this comment. Can you explain to me why this is a better way of saying this? Or can you, because sometimes, I think when people edit manuscripts, they'll just like change the writing. And I'll be like, oh, I don't understand if this is because I don't know how to write it properly, or like, is it a matter of style? And so understanding whether that feedback was like style, like, oh, just people have different style, or actually, no, like this is the incorrect way of saying things. Yes, that's the Steven Pinker book, Sense of Style. That's a really good book. And so that helped me understand whether I had to improve my English or people just write in different ways. And so that was that, and so that was the work I devoted for learning how to write. And in terms of, applying for academic positions, I think that the main thing is that, so I like having a trusted person that can just look over the English can be very helpful. And so make sure that all of your application materials go through this like trusted English reviewer. And that helped me a ton. Yeah.
You've spoken a lot about the effort you put in. Could you speak to us about things in the academic environment that were more inviting and enabling for you? Because of course, that's what we want to create, is an environment that helps people achieve their aspirations, not that puts additional barriers in their way.
Yeah, that's a good question. I mean, I think so I think in terms of like at least the language thing. Oh yeah, sorry. There we go. So in terms of the language thing, I know like a lot of universities have kind of like the Center for Language. It's mostly geared towards undergrads. And I don't know how helpful the help is because it's not very subject specific. And so I think having some sort of like within the department, you know, writing center or something like that. I mean, obviously this puts, you know, more work on the people who are willing, but like, I think language barriers are a big thing, and having some subject specific language helps. And because I think like language, like, right, it's not, you know, like one of my best friends, she's an editor, and like I can send her my things, but like she doesn't know anything about science. And so yeah, I think the other thing too, and this is, I mean, I think we have more about this, but it's preparing people for what could happen a little bit. So one of my first manuscripts that I submitted, like my advisor's first language is English, but her name is not very English sounding. And we got the feedback from that first submission. It was like my first author paper. It's like, oh, you should have an English speaker like read this. And I mean, it was like I cried, but I think like I wasn't prepared to know that people would be saying that just because of our names. And so at least even, you know, being prepared, like at least being prepared for the possibilities can help you, like, okay, people might say this, it might not be true, because then you feel like, oh, my English is not good enough, and it's like, well, you know, maybe that wasn't really the case.
I really appreciate that comment, Laura. I'm a senior editor at a journal, and I've written that sentence a number of times about the English in a manuscript. And I have always struggled with, you know, the paper could be crafted better. There are sentences that are getting in the way of the story. And how do I give that advice in a way that is not demoralizing is a really tricky one. And of course, it's a volunteer position, right? And we don't have the time and effort to try to copy edit individual papers. But I absolutely see that this is a barrier.
Yeah. I mean, I think like, at least with that particular paper, what would have been more useful for me is like having the sentences that are not clear being highlighted. Even if you don't tell me how to fix it, just tell me like, this is not clear. And that is more specific than saying like, this needs to be read by an English speaking person. Because I mean, first of all, my advisor is a native English speaker. So that was kind of, but then also like we did send it to somebody who was an English speaker and was like, oh, like this, it reads fine. It's just like this sentence, like this part is not clear. It's like, oh, okay, I can fix that.
Robin.
I'm going to do that dreaded academia thing because I'm not totally sure what my question is, but I really want to emphasize what you said about sort of how you reflect on things, because as a also relatively new tenure track person, I am now in a position where people are asking me to talk about being a woman in science. And I never really thought of it before. And now I'm supposed to talk about how I feel about it. So that thing that you said about I can't look forward, I can only look back and say, maybe that happened, but looking forward, I just have to do my best and assume that it's not impacting it. That fell so heavy on me that I just really want to make sure that we all heard it, because it's such a powerful and important thing that you really don't know why things are happening. And then later you look back and go, maybe that thing that happened, maybe wasn't just them trying to be supportive. Maybe that was kind of them being jerks or whatever. So it just, I feel like I want to shout that from the rooftops a bit. I really appreciate that you took this feeling that I had in my brain and kind of turned it into something I can actually say to people now. Yeah. So I hate the person who's like, I just have a comment, not a question.
I like comments.
Yeah, I really value that maybe that I'm also having that same experience. And that
I think collectively all of us are getting better language for describing our experiences. And I know over the decades I have now been in my career, we are talking about such different things. And just the ability to be open about the experiences we've had and that they were wrong has really shifted. And I hope that conversation continues and is amplified so that more and more people who are perpetrating the slights and the deliberate insults and attacks stop doing it. That more of us stand up to be counted and say, no, I'm here, deal with it, get out of my way.
Yeah.
I do have one question that's not related to that. You started in Columbia, then you were at McGill, and then you went to the other side of Canada and went to UBC and Simon Fraser, and now you're headed down to Los Angeles, and you're starting in a completely new space. All of those 4 places are very different culturally and linguistically. And how do you feel like things will change for you as you move south and move to Los Angeles?
That's a good question. So I actually, one of the things I've missed, like I've told you all this effort I've made to like improve my English, and then it's gotten to the point that I don't actually speak Spanish very much. And like here, I don't actually have the opportunity to talk in Spanish except to my parents and, like a couple of friends. And one of the many reasons why LA was kind of like this draw for us was like, and for me it was like, oh, it's an opportunity to like be reconnected with like my like Latino experience, and when we went to visit LA, it was hilarious because it felt like you had taken South America and Vancouver and mashed it together. And so like it's still North America, and it's still like it's organized somewhat. And then you go to some neighborhoods and like it just felt like I was walking back in Bogota. It was like the same street energy, the same food, everything. And I was like, oh, I miss this. And so that is one of the things I'm looking forward to. And, it's just like having a bit more of a mix between, all of these different cultures. I think that would be good. And the other thing, like I've actually made more of an effort to collaborate more with like Colombian scientists. And so both in science, but also in terms of like my outreach work as well. So I think those two things are helping me kind of like reconnect and like speak Spanish more often. And also the food. I'm looking forward to the food.
We're people first and scientists second.
Yeah.
Awesome. That's amazing. I'm so excited for you. I hope it's everything you want it to be.
Thank you. Yeah, me too. And yeah, the funding landscape is going to be interesting. Writing more grants is going to be interesting. But I think it's going to be exciting.
I'm eyeing the clock here. Does anybody have last questions or comments? Laura, any last insights for us?
I mean, I think that I just wanted to kind of like both re-echo what Robin said and you know, re-answered Shirley's question. And it's like, do the best you can and rely on your network. And I think those are the two things you can like worry about. Is like, okay, doing the best you can, but also, you know, making sure you rely on the people who are around you to like help you and like I definitely couldn't have gotten where I'm at without my network, both like my family, my friends, my mentors. And I think like one of the things that I realized too is like mentors, it's like everybody around you, like not just your advisor, but the postdocs around and the senior graduate students. And, you know, like one of the things I quickly realized early on was like, okay, maybe my advisor is very busy. Maybe she doesn't have like all the time in the world, but there's like postdocs who have more time. And then there's like senior graduate students who have more time. And like, so I'm going to bug them more. And like, I couldn't have gone it. Like I got to where I am because of everybody's support and help.
Yeah, thank you so much for the suggestions and your inspiring story. Yeah, I really
Thank you.
appreciate that. Thanks.
Thank you.
All right, everybody, gonna wrap this up. Please
Awesome.
join me in thanking Laura for a great talk and discussion, and very best wishes to you as you enjoy working at a big US private institution with all the weirdness that entails.
Yeah. Thank you so much. I really appreciate it. And thank you again for the invitation.
Oh, it's our pleasure.